for the rest of the time. And so it's the major immunodeficiency disease that's affecting people worldwide. So the origin of AIDS, it crossed the species barrier into humans in Africa in the 1930s. So it started out as a simian-based virus, like a primate, and then crossed the species barrier into humans 90 years ago. There was a patient who died in 1959 in the Congo, and that's the oldest known case. It then spread in Africa as a result of urbanization, of people moving into areas, and the disease spread that way. It then started to spread worldwide through modern transportation and unsafe sexual practices, and this was happening in the 70s and 80s. The first known case in the Western world, so basically outside of Africa, was a Norwegian sailor in 1976. Okay, in 1981 is when it really started becoming pronounced in the US for the first time. And the way they noticed it was that there were various cancers and bacterial infections that typically don't occur at high frequencies and don't cause death in people, were starting to cause these things. They were starting to cause death, and they were starting to see a spike in these types of infections in young homosexual men. Okay, so this was the first sign that there was something going around, and it appeared to be sexually transmitted. When they looked a little bit closer at these men, they saw that their immune function. So they took out some of their immune cells, and they did some tests on them, and they found out their immune cells weren't functioning properly, and they had low numbers of immune cells. And it took a couple years, but eventually they discovered the virus that they thought was causing this immune function, and they labeled that human immunodeficiency virus, or HIV. Okay, so here's a picture. So HIV infects cells through the CD4 receptor, meaning it infects mostly CD4 T cells, but macrophages also have CD4 receptors, so it can infect macrophages, but it solely infects cells of the immune system. So here's the structure of HIV. It's a very simple virus, not very complex at all. It's an RNA virus, so it basically comes with RNA. It comes with a reverse transcriptase enzyme so it's a reverse transcription, meaning it turns RNA back into DNA rather than normal transcription that goes from DNA to RNA. So it injects the RNA and these enzymes into a target cell. The reverse transcriptase then reverse transcribes this RNA to make DNA, and then that DNA gets incorporated into the host cell's genome. Okay. It has these glycoproteins, which are basically proteins associated with sugars, GP120, GP41. It has a small envelope, and that's basically it. So these GP120, GP41s, those are antigens that they can test for. So there's a lot of ways they can test for HIV infection. They can do PCR on your blood to see if they can find any of the HIV nucleic acid. They can look for the live virus. They can just do a test to see if they find any of these GP proteins in your blood. So this is how infection happens. So you have, let's call it a CD4 cell that has a CD4 receptor. These glycoproteins bind to the CD4 receptor, causing the viral envelope to fuse with the cell membrane, and that allows the nucleic acid and the reverse transcriptase to be inserted into the cell. This is basically what I was just talking about, but you have your viral RNA with the reverse transcriptase. That viral RNA gets converted into viral DNA by the reverse transcriptase. That viral DNA then gets inserted into the host cell DNA. That DNA then gets transcribed into mRNA and gets translated into protein, just like the DNA of the normal host cell, except it's making viral proteins. Those viral proteins then can come together to form a new virus. And this is the cycle of the infection. The important thing to notice is that because it's a provirus, it's DNA that gets incorporated with the host cell DNA. It can stay latent, meaning again it doesn't have to go into the lysogenic cycle. or into the lytic cycle, it can stay in the lysogenic cycle and just remain in the DNA. So you can get rid of all the active virus in the bloodstream, but if you stop the medication, at any point in time, the DNA can start producing more virus again. So it's very hard to get rid of because of that. Here's just a full schematic of how that works. This is basically just step by step. There's different subtypes of HIV. I'm not going to go into all that, but depending on what geographic location you're looking at, one subset, or they're called clades, one clade might be more prevalent than another. So clade B would be what you find in North and South America and Europe. Clade C, that's more, now it's more than half of all HIV infections are what you would find in most of Africa and India and parts of China. And clade E would be what is typically found in Southeast Asia. So these are the stages of HIV infection. So phase 1 is basically asymptomatic or chronic swollen lymph nodes, because you have an active ongoing infection that's not being cleared by the immune system. And the lymph nodes are where the cells of the immune system undergo their division, so you're just constantly having cells of the immune system dividing, and so your lymph nodes are constantly swollen. Okay, the initial infection, the symptoms aren't much more than the flu. Phase 2 is when it becomes symptomatic. You start to get early indications of immune failure. You start to feel weak, tired. You start to get ill more often. You know, you just get the common cold more often. You get the flu more easily. And then phase 3 is when you become what they call seroconverted, and you become from just HIV positive to an AIDS patient. And basically that is based on how many T cells you have in your blood and how much active virus you have in your blood. So once you reach again, because HIV is killing your CD4 T cells and your macrophages as it's replicating. So once your CD4 T cells reach a limit of below like 200 cells per microliter, and your viral load goes above, I can't remember the threshold, but when you hit these two levels, you're considered seroconverted to AIDS. Okay, so here's basically how the viral load and the CD4 T cells look. So let's see if I can help interpret this. All right, so the blue line would be the amount of virus that's in your blood, and the red line would be the number of CD4 T cells you have over a point of time. So this goes up to 10 years in this graph. So you get an initial infection, okay, at point 0, and you start to get a ton of virus in your bloodstream. That's because the virus is basically replicating faster than the immune system can keep up with it. So you have a lot of active virus. Okay. Eventually, this causes a spike in the amount of T cells that are now trying to fight the virus, and as the amount of T cells goes up, it's able to make the virus basically go down to almost undetectable levels because it's actively fighting it. Okay, so you can see within the course of a year, you've gone through high viral load, now you're back down to pretty much baseline level. It's not zero, it's between 1,000 and 10,000 per milliliter active viral loads, but it's much lower than the 10 million it was at its peak. And basically what's happening is it's just replicating at a very low level, and the immune system's trying to fight it off, but as it's replicating, it's killing cells of the immune system every time it's replicating. And so your CD4 T cell counts start to go down, 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 down slowly. And again, once they reach the point of under 200, it's basically a critical level where your immune system is basically completely shot. Okay, because again, you need the CD4 T cells to help activate B cells and to help activate the CD8 T cells. Okay, the CD4s are the helper T cells. They produce those chemicals that let the other types of cells be activated. And if you have no CD4 T cells, basically it means you have no CD8 response and no B cell response. So it's basically totally decimating the immune system. Once your T cells reach such a low level that you basically have no active immune system, the virus is basically able to start to replicate undeterred by the immune system, and it basically takes off exponentially. And so usually when you reach this point without medication, it's a short amount of time before death. So typically, it's not HIV itself that kills a person. It's the fact that their immune system is so weak that they get these other diseases that typically can easily be fought off by the immune system. But because there is no immune system, these diseases start to kill people that are infected. Okay, so there's CMV. I can never remember the exact number, but something like 95% of people are CMV positive. So in this room, there's maybe 1 or 2 people that aren't CMV positive. It's a viral infection that really has no symptoms. It's easily controlled by the immune system. And however, in someone that has AIDS that has no immune system, CMV basically takes over and can actually kill the person. What else here? Tuberculosis. So these are things that typically herpes simplex virus, things that our normal immune system would be able to maintain. So there are a subset of people that actually can be infected with HIV and never progress to full-blown AIDS. Okay? So one way is to have a mutation. So if I can go back a few slides here. Okay. So in order for the virus to adhere to the host cell, it binds to CD4 receptor, but it also has to have this co-receptor that it binds to. Okay, CCR5 or CXCR4. Okay? So there are people that have a mutation where they don't produce CCR5 or CXCR4, and basically it prevents their cells from being infected by HIV. There are people that just get a very strong initial immune response by their CD8 T cells that basically completely rids the system of the virus before it can become weak. Then there are people that are called long-term non-progressors that basically don't have CCR5 mutations, don't have a particularly strong immune response, but they never progress to full-blown AIDS. Probably the most famous case of this would be Magic Johnson. Okay, he's been HIV positive now for, gosh, I was going to say, I think it was 10. Yeah, that would make sense. Yeah. So what is that, 20-something years? And he's fairly healthy. Mechanism is not known. And so there are, I don't want to give a percentage, but definitely less than 1% of people that get HIV infection that for whatever reason just never get full-blown AIDS. Okay. Again, these are the ways that you can tell if someone's HIV positive. You can look for antibodies against HIV. You can look for proteins against HIV. Like I said, those GP proteins, the glycoproteins, you can do what's called Western blot and see if there's any of that protein in the blood. You can actually just take plasma and do a PCR reaction and amplify viral DNA, and if there's any viral RNA, then you can detect it that way. So the way HIV is transmitted, it can survive 6 hours outside a cell. So just on in a droplet of blood that might be on a surface somewhere, HIV can remain alive for about 6 hours. Inside the cell, it survives less than 1.5 days, but that's plenty of time in order to replicate and form more viral proteins, particles. Infected body fluids basically are what transmit HIV. Sexual contact, obviously, breast milk, transplacental infections of the fetus, blood contaminated needles, organ transplants. Because again, so anytime, obviously, they do a blood donor transfusion or an organ transplant, obviously they test it for HIV. Artificial insemination, again, blood transfusion. So worldwide, again, not just in the US, but worldwide, 85% of HIV transmission is from heterosexual intercourse. So there's still a stigma, I think, associated with it that it's mostly affecting homosexual males, but it's actually 85% of the infection is transmitted by heterosexual intercourse. Injected drug use is another big one. Sharing needles. And 42% of everybody that's infected is a female. So here's the, I don't know, what year is this? Does it say? 2010 maybe. So it's fairly recent. This is the worldwide distribution of AIDS and HIV. You can see by far the biggest cases in sub-Saharan Africa. there's a handful of reasons. I'm not going to get into all of them. But the main ones are that there's just a lack of education. There's not a huge belief in contraception. And there's not a lot of medication. Medication for this is very expensive, and it's hard for them to afford it. So you can see, again, Sub-Saharan Africa, the second biggest section would be in Southeast Asia. But even in North America, a lot of people kind of think that HIV is more or less not as big of an issue as it was, and it might not be as big of an issue as it was maybe in the late 80s, but there's still well over 1 million people that are HIV positive in the U.S. So preventing transmission of HIV or AIDS, obviously the use of condoms, sterile needles, these are all kind of common sense things that now, because it's been beat into our head a little bit, this wasn't always things that, this is things that people took for granted only maybe 30 years ago. Circumcision, there's actually some studies to show that circumcising males actually helps to prevent the transmission of HIV. Healthcare workers using universal precautions, so always anybody that's working with blood, wearing gloves, gowns, masks, goggles, not recapping needles, so preventing a healthcare worker from sticking themselves with a needle. So one of the main questions that people have is that if it's such a big deal, it's a virus, we have effective vaccines against almost every virus, why don't we have an effective vaccine against HIV? And believe me, billions of dollars have been put into it, and we're just really not any closer than we were a decade ago. So these are the 5 main reasons why vaccine difficulties that are specific for HIV. HIV is a highly mutated virus. It mutates all the time. So any vaccine that could be given at a certain time, HIV would have the ability to undergo a mutation quickly so that it could evade the immune cell. There's these different clades, like I said. So each clade has slightly different genetics and makeup, so you couldn't just have a single universal vaccine. You have to have different vaccines for the different clades. The antibody binding sites are hidden. Okay? So basically what that means is that most of the virus is inside of cells. It's not in the bloodstream. So if you could get a vaccine that would give a good immune response, give good antibody response, that antibody could get rid of the virus that's in the serum, but the virus that's inside your host cells is basically hidden to the antibody. The antibody can't recognize it if it's inside the host cell. So that's one issue. The infected cells are not susceptible to cytotoxic lymphocytes, okay? Because it's infecting your own immune cells, and those immune cells are not susceptible to cytotoxic lymphocytes for the most part. And it's a latent virus, again, which means the DNA can just remain incorporated in the host cell DNA for years and years and years. The vaccine could work and get rid of all the active virus in your system, but it can't get rid of the incorporated DNA that's been incorporated into the host cells. So the therapies that are currently being used, nucleoside reverse transcriptase inhibitors. Again, reverse transcriptase is an enzyme that the HIV needs to turn its RNA into DNA, so it can be incorporated into the host cell DNA. So if you can inhibit that enzyme, it has some ability. Protease inhibitors and fusion inhibitors, I'm not going to get into it too much. So usually people that have HIV are on HAART, which stands for highly active antiretroviral therapy. So it's a combination of these different inhibitors. And they'll typically give somebody a cocktail of different combinations of inhibitors, see how they do for a few 6 months, and then if it's not looking good, they'll change it up a little bit. So they try these different cocktails over time. And again, they're pretty toxic, so they cause toxicity in the patients, but it's better than the alternative. Okay? Any questions about that? All right, because we have a little bit of time, I'm going to pass out the assignment for the disease project and talk about that for about 5 minutes, and I'll let you guys go on your break.
Thank you.
All right. Everybody has one? All right, so this will be the final project. It'll be one of the last accumulations of points you have for the class. This in the lab notebook, and the final basically will make up the last 400 points of the class. Okay, so 40% of your grade will basically be in the last week or so of class. Okay, and this will be 100 points. So you'll choose any etological microbial agent, okay, that causes a nationally reportable communicable disease that has been in the news recently. So I want to highlight that. I don't want you to do a report on polio in the U.S. that was eradicated 70 years ago. Okay, I want something that's been in the news, preferably within like the last 5 years. Okay, even more preferably something that's happened in the last month or two. Okay, and I'll show you where you can find some of those resources. Okay. You'll give background on the agent itself, so on the bacteria or virus, disease it causes, and what the recent development is. And you'll do that in a 5 to 10 minute oral presentation that you'll give to the rest of the class. Okay. The oral presentations will be given during lab during the last 2 weeks, and I have dates below. So it'll be a written report. That'll be 3 or 4 pages, and then also an oral presentation. Okay, and they'll go together. So the report, you write 2 paragraphs on the significance of the microbe itself. So a little bit of background. Again, you'll include appropriate references. 2 paragraphs on the background on the disease itself, and you have to include references there too. And then 2 paragraphs on the new development of the disease. So what is that recent development? Okay. And you want references there too. So every section should have at least 1 or 2 references in it. Okay, your report should include graphs and or pictures. So, and I'll get into this. Some of the research you have to do will be from primary research papers, and those papers typically always have some kind of table or graph. So you can include that in your report to help make your point. Citations should be included throughout. Here's the important one. At least 5 total references must be used in the report, and 2 of these references have to be obtained through PubMed. Okay. So if you guys saw on the lab schedule, there's a take-home assignment in the lab. It is exercise 29. It tells you how to use PubMed and how to get primary research papers off PubMed. So you do that exercise in your lab book. They'll teach you how to do PubMed and then use it for your report. Okay, so 2 of the 5 references have to be from PubMed. And PubMed will give you the primary research articles. Okay. Formatting between 3 or 4 pages. You have to address all 3 of the sections above. 4 if you include the citations part. Include a cover sheet, and blah, blah, blah. Okay. If you look at the back, it talks about the oral presentation. So, I'm sorry, the report is due December 4th. It's the Wednesday before the final. Okay. The oral presentations, because there's so many of us, we won't do them all on one day. We'll split it over 2 days. Half of them will be done on the 4th, maybe more than half, but some of them will be done on the 4th, some of them will be done on the 9th. But everybody's report, regardless of when you present, is due on the 4th. Okay. We probably won't get done with everybody's oral presentation on the 4th, but you should be prepared to go on the 4th, because if everybody does closer to the 5-minute mark, we actually may be able to get through everybody on the 4th. So come prepared to present on the 4th. We may not get to you. I'll basically have a sign-up sheet. Some people want to go first, some people don't. I'll basically have a sign-up sheet that people can sign up for to determine the order in which they'll present. My guess is we'll get through 15 to 18 on the first day, and then the remaining 8 to 10 on the second day. But everybody should be prepared to present on the first day. Should be 5 to 10 minutes long, And that'll be part of your grade will be based on your timing, that you're not under 5 minutes, you're not over 10 minutes. You want to provide background on the microbe and the disease. So you basically want to do an oral presentation that summarizes your report, background on the microbe, the disease, and talk about the new development. It should be some kind of interactive and have some kind of visual aid. So the visual aid could be PowerPoint, you could use whiteboard, you could use handout, anything else creative you can think of. And then at the end, you want to be sure to ask for and be prepared to answer questions from the audience. Okay? The point breakdown. So if you look at it, if you add everything up, the report is 60 points and the oral presentation is 40 points for a total of 100 points. Okay, any questions? All right, so I do want to show you guys one good resource if you're trying to figure out what you want to do. Oh yeah, one important thing. I say this every semester and people either don't listen to me or forget that I say it, but you don't have to ask me if you can do something, okay? So if people come up to me, can I do this disease? Can I do this? You don't have to ask me, okay? If 5 of you guys end up doing the same disease, then 5 of you guys end up doing the same disease, okay? You don't have to reserve it and run it by me and then nobody else can do it. It's just up to you guys, okay? It's up to you guys to just follow the directions. You don't have to, if you want to ask me if it's an appropriate disease, you can ask me, but don't just say, I want to do this, is anybody else doing this? I want to do this one. You don't have to ask me, just do what you want to do, okay? Obviously, the more, you don't want everybody to do the same one, so do a little bit of research and try to find one that's maybe not as, broad that you think everybody else might be doing, but it's up to you, okay? So let me show you a good resource. Trying to think of how I Googled it last time. I think it was CDC recent outbreaks. I think if you Google that, yeah. So that'll basically give you the current outbreaks, okay, or anything recent. Okay, so there's a salmonella outbreak from Foster Farms chicken that's happening right now. You could do that. Okay, right there, it's salmonella, so it's a microbe, it's a bacteria. And there's a recent development. They're finding it in Foster Farms chicken. So you would do a couple paragraphs on salmonella, a couple paragraphs on the disease of salmonella food poisoning, and then a couple paragraphs on the recent outbreak and get some data there. Okay, another salmonella. Hantavirus at Yosemite. Okay, that happened last August. That's a good one. Pertussis in Washington, April 2012. All right, so this is just a few. You can do international ones. They don't have to be U.S. based. Ebola outbreak in the Democratic Republic. Whatever you guys want. Okay, so this is one resource. Something else you can do just every day. You go to CNN.com and check the health section of CNN.com. You can do some stuff there. The health section of Yahoo or anything. They'll have some things that come up. So do a little research. Try to find one you find interesting, and that's it. Yeah.
So recent, how many years? Like in the past 5, 10?
I would like to keep it within the last 5, but if you really want to do one that is 10 years, that's fine. I'm trying to avoid, like the ones that are, I don't want someone doing the mumps, No one gets the mumps anymore. Okay. All right, any other questions? No? All right, so you guys have a break. We'll start lab. We have a fairly short lab today. So we'll start lab around 7. So just be ready to go.